this textbook you can find in internet and use for your preparing. About our topic. So we must revise that nucleus present only in eukaryotic cells. This structure is a membrane bound structure, and inside we can find DNA, molecule that contain genetic information about the cell. Functions of nucleus. They are. So they take part in division, cell division, stores the hereditary information of the cell, regulates metabolic processes of the cell. All processes need enzymes. All processes in our cell, they must be catalyzed by enzymes. All enzymes contain protein part or protein at whole, and all proteins are synthesized in matrix of DNA. So next slide. Can I change this slide? Yes. Thank you. Structure of nucleus. So nucleus bounded, we said, by a double membrane, and we can find outer nuclear membrane, inner nuclear membrane. Between them, we can see nuclear space, inner nuclear space. I'm sorry. And this envelope is penetrated by pairs, all nuclear complexes. Outer nuclear membrane is connected with the membrane of endoplasmic reticulum, root endoplasmic reticulum. You can see ribosomes and that located on nucleus envelope and on endoplasmic reticulum. So can I change this slide? Thank you. And you can see that nuclear envelope consists of two membranes, more better in this photo, and it's penetrated but porous, which form holes between the cytoplasm and nucleoplasm. Each pore is surrounded by a nuclear complex that consists of 8 large multiprotein granules. So it is very difficult complex structure that include a lot of proteins with their own functions, and it will be channel for transport RNA in cytoplasm and substance for creating RNA from cytoplasm in nucleus. Can I change this slide? Thank you. Proteins of the Proteins of the inner nuclear membrane form a nuclear lamina. Lamina very important structure inside nucleus because it helps to begin mitosis because they takes part in destruction of nuclear envelope. So lamina, it is a fibrous network that located near with the inner nuclear membrane. Can I change this slide? Thank you. Thank you. Inside nucleus, we can see structures that are gammagenous. It will be chromatin, complex of DNA and proteins, and nucleoplasm, like cytoplasm. This is a jelly-like solution that contains proteins, lipids, carbohydrates, RNA, nucleotides, and enzymes. You see nucleolus in the center. This is not membranes bond structure. This is complex of DNA of chromosomes that located so far, so near with each other. So nucleolus has no membrane, and this part is nuclear organizers. Contain information about rRNA, tRNA, R or ribosomal RNA, mostly information about them located in this part. So can I change this slide? Thank you. Chromatin consists of, we said, DNA and proteins, histone proteins, and non-histone proteins. And also it will be RNA that will regulate levels of compactization during life cycle of cell. So you see the first level will be nucleosomes level, after solenoid, interphase chromatin, and during mitosis, metaphase chromosomes. We will discuss it in the end of the lecture or in the next lecture if will not if our time will over today. I'm sorry for this. So nucleic acid, DNA and RNA. Nucleic acids are bipolymers. They contain nucleotides. Every nucleotide consists of 3 parts: nitrogenous base, 5-carbon sugar, and a phosphate group. Can I change the slide? Thank you. So we know there are 2 groups of nitrogen bases. They are pyrimidines and purines. The first group, cytosine, thymine, and uracil. Cytosine only for DNA, uracil only for RNA, and the second group, adenine and guanine. This is easy. Thank you. So adenine, guanine, and cytosine, they are both in DNA and RNA. Thymine is only found in DNA and uracil only in RNA, we said it. So, complex of deoxyribose not contain oxygen atom in, in carbon 2. This is difference between them. I try be more fast because lecture is long. So nucleotides are linked to each other by phosphodiester bonds from from 5' end to 3' end. So from 5 atom of carbon to 3 third atom through the phosphate group and oxygen. And always process of synthesis will be go from 5' to 3'. Is it clear? Very good. Thank you. So the two free ends of polymer differ from each other. At one end always will be phosphate group. In the end, in the 3' end always will be hydroxyl group. And DNA. DNA molecule consists of two long chains or strands. They are organized like double helix, and they are connected by hydrogen bonds through the nitrogens that are. I'm sorry, I forget. I forget the word. In Russian, forget, and in English, forget. Okay, I will remember after. So, nucleotide consists of three parts, we said: pentose, nitrogen base, and one or three or two phosphate groups. They are antiparallel and complementary. This word. So, adenine always complementary is always complementary to thymine. Cytosine to guanine. Yes, of course. This, Ahmed, this slide. Thank you. Спасибо. So this means that opposite 5' always will be 3'. opposite adenine always will be thymine, and so on. Complementary. Sugar phosphate bonds are located outside the helix, and nitrogen bases are connected inside the helix by hydrogen bonds. Two hydrogen bonds between adenine and thymine, and three between guanine and cytosine. The next slide. Yes? Thank you. And if we have these sequences, for example, AGGT-CCG, so according to the complementary, will be complementary trait. I'm sorry, my English today very bad. Will be opposite adenine thymine to guanine cytosine and so on, and you see opposite 5' end will be 3' end, but RNA synthesis will be only in one strand, only from matrix that beginning from 3' to 5', because new RNA strand must begin from 5'. So the first strand that we see in this slide is only coding strand and takes part only in duplication of DNA, but not in biosynthesis of protein. Is it clear? Thank you. The next. Antiparallelism. Next slide. We said about this that these strands are antiparallel, have antiparallel directions, so opposite 5' will be always 3' end. And the third rule, very important rule, that amount of purine, we will talk about coding RNA later, okay? If you will not have answer, ask me again after, okay? So amount of purine, thank you, purine nucleotides always equal to amount of pyrimidine nucleotides. The fourth rule, DNA is right-handed double helix, and nitrogen bases are located inside this helix. Next. Yes, can I change the slide? Thank you. Role of DNA, biological role. So we said this is storage of information, replication of DNA, and taking part of in mitosis, and passing the genetic information from cell to cell, and regulation all processes in cell. We said it in the beginning. DNA molecule usually carries several hundred or more genes, but not all of them are active in each cell of multicellular organism. In one cell working one group of cells, in another will be another genes. When cell division occurs, DNA molecules are copied and passed from one generation of cells to another. And genetic instructions that an organism inherits from one parent are called gene. So this gene, this will be the unit of our genetic information. This unit has beginning and the end. That's why not all information will be expressed in each cell, because we have small units, genes. They are different, they have different number of nucleotides. They may be large, about 2 millions of nucleotides. They may be small, they may be specific for several cells. They may be common for all organisms. We will see this and more of this during second year of our education, during studying genetics. But now we will talk about simple characteristics. And this information that encoded in genes that contain information about proteins, this information is so small if we compare with all DNA sequences that are located in our DNA. And in seventh Brins and Davids, they discovered a fundamental feature of eukaryotic genome, that we have different sequences according to their repeatability. And there are three types, now we know three types of sequences according to this. Unique sequences, sequences that can be repeated in our genome, and it will be medium repeatability sequences and highly repetitive. Unique sequences contain information about our proteins, and they are not, we said they are not big copies, about from 1 to 10 copies, and it will be only 20-30 percentages of our genome. Other 17% of our genome will be repetitive sequences. So medium repeatability sequences, they contain copies from 10 to hundreds, and they contain information about ribosome, transport RNA, histones, and immunoglobulins. Highly repetitive sequences do not contain information about any proteins, but their function, we don't show about their function in this time, but we think, genetics think, that their function is regulation of biosynthesis proteins, regulation of our our general mechanisms in our organism, regulation of our life. So scientists think that one variant of these repetitive sentences can begin our embryogenesis. So if the sentences will absent, our pregnancy will not begin. So if you will study well, we will talk about in this during genetics. So I hope that you will be good in first year education, and we can speak about difficult problems of genetics in the second course. I hope. Next slide. So, thank you. We see our genome contain two variants of sequences, we said, In this slide, only 20% of genes, yes, because other 10% will be pseudogenes, but it's not material for first course. About gene, we said it functional unit of heredity and contain information about protein or RNA. The next slide, yes. Thank you. And eukaryotic gene, very difficult in structure. It contains three parts, main parts, two of them in the end of genes, in the ends of genes. They are regulate parts that have regulation function. They are promoter and terminator. You see these parts green and blue. In the middle between them, structural gene itself that contain two variants of sequences. We call them exons and introns. Exons, they are dark in our scheme. They contain information about proteins, and they will be translated during translation. Introns in eukaryotic cells present only in DNA, and they do not contain information about proteins. They very important for regulation of transcription and present only in first period of biosynthesis in transcription. Such as promoter, this part important only for beginning of transcription. So most part of promoter will be present only in DNA, and only last small part will be transcripted and after that translated during biosynthesis. So the next slide, yes. Thank you. So genetic code is a way of coding information about protein structure in DNA. How it will be? Not before about characteristics of genetic code. The first, every triplets may encode only one amino acid. These codons do not overlap. But we must remember that inside prokaryotic cells, overlapping may be. Now we talk only about eukaryotic cells. Here overlapping impossible. Next slide. You see in this slide overlapping. The first codon ATG, the second codon will be G again, AA. The next AAG. So one nucleotide will take part information of two nucleotides. It's possible only for prokaryotic cells, not for eukaryotic cells. You see on the right side, ATG, only one nucleotide in one codon. The next nucleotides will take part or information the next nucleotide. They will not double. Thank you. So genetic code, again, about what again? So in our cells, eukaryotic cells, one nucleotide in this sequence will be used only once to formate codon. ATG, you see the first codon, the next AAA, the next GCG in our slide. The same sequence in prokaryotic cells. You see that in first nucleotide came ATG, the second nucleotide codon. What again? About eukaryotic cells again? So ATG, the first nucleotide in eukaryotic cells, and only one nucleotide, only one codon. The next codon will be with another nucleotides, AAA. Before this, you see in the light side of our slide, ATG, and this G will take part in another nucleotide. So in eukaryotic cells will be only 3 codons. In the prokaryotic cells, we can count 1, 2, 3, 4, 5, 6, a lot of codons. Okay. Okay. So this is our DNA. Nucleotides. RNA polymerase begin creating of RNA, so it will be DNA. In eukaryotic cells, it will be one, two, three, the same, yes? One nucleotide, one. The next, yes? So we had 6 nucleotides, and after that we have 3 nucleotides. So will be 1 amino acid, 2 amino acid only. Eukaryotic cells. Prokaryotic cells, the same sequence will be 1, 2, 3. Polymerase again 1, 2. In the opposite, so it will be 1, 2, 3. Again 1, 2, 3. So in this sequence of DNA, where was only 6 nucleotides, we have 1, 2, 3 codons. So it will be 1, 2, 3 amino acids. Do you understand? Thank you very much. So we can continue, yes? Thank you. Now I need to find my presentation. So the next code is I'm sorry, deciphered in a single direction without stops. So again, we talk about only eukaryotic cells, about eukaryotic cells. And code-specific, every codon only codes a single amino acid. But amino acids may be encoded by multiply codons. These protect our DNA from mutations. When one nucleotide may be changed, we have the same amino acid during this characteristic, during the degenerate characteristic. Is it clear? Thank you. So genetic code properties. The next. Universal. All cells have one same code. But again, prokaryotes may have small differences and our mitochondria have some differences. There are several specific codons. One codon for the beginning translation, start codon AUG, and stop codons for finish of translation UGA, UAG, and UAA. Next slide. Thank you. Protein biosynthesis. The linear order of bases in gene determines the amino acid sequence, and it will be primary sequence of protein. So we begin. There are two main stages of biosynthesis of protein. They are transcription and translation. They always present during biosynthesis. In prokaryotic cells, always. In eukaryotic cells, always. But in eukaryotic cells will be two new cells also. Two new stages also. We begin from transcription. This will be RNA synthesis on DNA matrix. And transcription beginning from the promoter. You see a small part of promoter after transcription we have in the 5' end. And a little part in terminator. It was saved. After transcription, all exons and introns will be in RNA sequences. So this also not mature RNA because it has introns. So after the transcription, we have premature mRNA that need have modifications. About structure of gene in DNA, we said main three parts, promoter. For beginning, RNA polymerase binds to it structural gene that include exons and introns and terminator region where transcription stops and translation stops too. Is it clear? Okay, thank you. This understanding very important to understand all processes in our organism. And again, not so easy all. You see, promoter is very small. And after promoter, before gene, there are present another regulative parts. It will be enhancers and another elements They are very important to the function of gene, but they are not part of gene. So again, our gene includes three main parts: promoter, you see light blue part, terminator, dark blue part, between them, structural gene with exon and introns, and before gene, regulative parts, for example, enhancers or silencers. These parts very important for formation transcription complex, for beginning of transcription, for initiation. Is it clear? Thank you. Thank you. So again, because even Russian students in the second course cannot tell me what is the main structures of gene. Again, they are promoter, terminator, and structural gene. Promoter, we need to connection with RNA polymerase for beginning transcription. RNA is usually a single strand, and every matrix molecular genetic process can only go from 5' to 3' end. For revising, yes. Stages of transcription. They are three. Initiation, elongation, and termination. I have a question. You write your name until now, 11 with 30. You just come in our lecture only now? So initiation, this very important part, very complex. About what, Abdullah? It was not for your question about two students who write here their names. So after 14 minutes of lecture. So initiation, very complex stage. Main event in this stage will be binding of RNA polymerase to promoter. Elongation, the next stage. This is synthesis of RNA, and the end, termination, ending of synthesis. The next slide, yes? Thank you. So RNA polymerase is not small molecular, very big molecular. And this part in promoter not take all this molecular. So you see a white arrow, it will be locus of promoter. Yes, all other is a part of gene, structural gene. In prokaryotes, a translation starts immediately after transcription, we said, because they have no introns. In eukaryotic cells will be post-transcriptional modifications, and they will include splicing and modification of ends. This is because eukaryotic genes have exons and introns, and introns we don't need after transcription. They must be removed from sequence. Next slide. Thank you. So that's why that they have introns. Primary mRNA in eukaryotic cells are processed after transcription, and after that will be functional mRNA. Stages. There are two main stages: splicing and modification of 5' and 3' ends. Splicing includes two stages: removal of introns and connecting of exons. Remember this, please. It's very important stages during our biosynthesis of proteins that may regulate formation of our proteins and may be reason of our diseases. So the next slide, yes. About processing. The first, removing of introns. You see red exons, blue introns. In our organism, there are enzymes that called endonucleases. They will remove these introns, and another enzymes, ligases, will connect exons together. During practical lessons, we will discuss this process because this is not easy. That students who are not in Kirov, you need read big textbooks about this. And if you have question about this process, when you will be in Kirov, ask your teachers. So for removing in our organism, in our nucleus, there are found complex structures that we, I don't know your group, Ahmed. There are complex structures that we call spliceosome. Spliceosome include small nuclear RNA and enzymes and the nucleases that will remove these introns, and after that enzymes ligases will connect exons together. Connection may be different, and this again you must ask in your lessons from your teacher. I don't know your group. Tell me your number, I will say. No, it's not my group, I'm sorry. No, it's not my group. No, I'm sorry. I have only 104 and 106. Yes, Amir, thank you. So, translation, protein synthesis. The next stage. The first was transcription, the second was translation, processing, and the third translation. About your teachers. First group, Anton Evgenievich Zubarev and fifth group, Anton Evgenievich Zubelov. Fourth, second, third, and seventh, Vladimir Ilyich. So translation. It may be only with the help of tRNA, transfer RNA, that has in the beginning anticodon that will be complementary to codon in matrix RNA, and in the past part, codon for connection with amino acid through the phosphate, through the carbon group. So this transfer RNA must connect it with mRNA, and first always will be methionine that will be connected with the start codon UAG. All proteins synthesis in our organism will begin from methionine. Sometimes this methionine will be removed after, but always it will be first amino acid. Initiation of translation, this assembly of initiation complex that consists of small ribosome subunit, AUG start codon, and methionine tRNA. When this complex will be created, large ribosome subunit will connect it with this complex too. So you see three main center in this ribosome, but we're interested only in two of them, P and A. The first center, it will be P. For first tRNA, the second RNA will connect it with A center always. P center is an important center for formation of peptide bonds. Acceptor center for binding of tRNA, next tRNA. Can I change the slide? Спасибо. Thank you. And this scheme may help you understand the movements of ribosome. So this jumping. The first you see in the high part of our scheme, methionine connected with the P center. After that, next, tRNA connected with A center. After that, in the right side, you see formation of protein peptide binding. And during our lesson with group 104, I said that there are no enzymes, but it's not like for your next course, it's better if you will know GTFase enzyme GTFase that help to connection of tRNA. So we need energy of ATP or GTP molecules. If you don't understand, please ask me again during next lesson, okay? Group 104. Nobody here. Amir, okay? Other students, please. Thank you. Other students, please ask your teacher. This difficult to show during lecture, we need discuss. So, in the right side, you see connection of two amino acids. After that, all amino acids we see in A center. P center is empty in the left. P center became, the first was A center. So the first the first tRNA go out. That center that was A became P, and new A center is ready. Do you understand? I'm sorry if not, but ask your teachers. Read textbooks. Okay? Thank you. Next slide. Termination is a stage ending of translation. And it's possible when we see stop codon in A center, in acceptor center. So after translation, we have primary protein structure. After the formation of native three-dimensional structure of the protein takes place with endoplasmic reticulum. And this process, formation of secondary, tertiary, and quaternary levels of protein, we called folding. Remember this name, folding. This process go into endoplasmic reticulum. Next slide. Can I change the slide? Thank you. Thank you. So, at any given moment, only the genes, the products of which are needed in the cell, in this cell, in this certain cell, yes, are active. That means that there is the regulation of gene expression. For regulation, we can begin from simple example, regulation in prokaryotic cells. And for example, we will use bacteria that produce three enzymes for regulation lactose metabolism. There are not only genes, structural genes in prokaryotic cells. There are also present regulatory genes. They produce proteins that can stop or begin transcription of other genes. So expression of these regulatory genes will be always, but they can stop or begin expression of other genes. And the expression will be only in time that they need for bacteria. These genes may be responsible for expression of several genes. For example, lactoseperon will regulate 3 genes, and a group of these genes will formate genetic regulation unit, or operon. Is it clear? Thank you. So the next slide. Lactoseperon include, like other operons, include promoter, operator, structural genes, in this case 3 structural genes, and terminator. Promoter, like usual, is a start point of transcription. Operator is placed for regulation by regulated proteins. Structural genes without introns and the terminator, place for stop. Is it clear? Thank you. The next. So this regulatory gene located near with the operon. And when lactose present in environment, this regulatory protein will be connected with this lactose. Without lactose, this regulatory gene will produce proteins that will connect it with operator. This picture. So you see regulatory gene near with the operon. Operon contain promoter, operator, and in our slide you see the first structural gene, gene Z. Regulatory gene always produce mRNA, you see purple, that will translate it into protein active repressor. Always, all time of living of bacteria. You see red structure. This red structure is very close in structure with operator. And this structure, this repressor, can change shape of operator. So when RNA polymerase will come to promoter, it can not move along the gene. It will be stopped by repressor. Is it clear? Thank you. So the next stage, when lactose is present in environment, so this purple mRNA from regulated gene will produce active repressor, but this repressor more close in structure to lactose than to promoter, to operator. And now RNA polymerase may move along the genes and create 3 proteins. You see galactosidase, in English, very difficult for me. I'm sorry. I will not read these names. So three proteins for metabolism of lactose. Bacteria begin use lactose for growing and replication itself. When lactose will finish in environment, this repressor will connect it again with the operator, and these enzymes will not be synthesized until lactose will not again in environment. Is it clear? Yes, thank you very much. So it's very easy variant of regulation of biosynthesis, and it may be only in transcription level. So remember this, prokaryotes or bacteria can regulate their biosynthesis of proteins only in one level, in transcription level. And for this they have regulator structure, operon, regulator unit. In eukaryotic cells, regulation will be in all levels of biosynthesis, in transcription, in level of processing, in level of translation, in level of folding, and also before transcription, pre-transcription level, and in whole organism level, for example, by hormones. So very difficult. and operons are absent in eukaryotic cells. So eukaryotic cells, they have no operons. Every gene regulated by multiplied regulationary genes and sequences, yes sir, enhancers are lesser before genes and regulated proteins, for example, transcriptional factors. A lot of regulatory genes and sequences. You see enhancers for beginning and silencers for stopping of transcription. Is it clear? Thank you. The next. Regulation occurs on every level of realization, we said. Excuse me. Of hereditary information. In our slide, I said only 3 of them. For first course, it's enough. Remember them. So it will be level of transcription, level of translation, and post-translational level of folding. Can I change this slide? Thank you. The first level, transcriptional level. The first must be initiation, you remember, yes? Connection of RNA polymerase with the promoter. For this, we need very difficult processes. They are complex. We need transcriptional factors. Usually this group contain about 8-10 proteins. They are needed for RNA polymerase must see where promoter on gene and the transcriptional factors will show here. This is promoter. Go to here, go and connect with promoter. So transcription factors show RNA polymerase where is promoter. Transcription factors very important to connect RNA polymerase, part of RNA polymerase to promoter, because without them RNA polymerase will not connect with promoter well. So it may be very not closely contact, that not lead to synthesis of RNA. This complex include distal control elements like enhancers. They help to attach RNA polymerase to promoter, like transcription factors. So if we will have no transcriptional factors, RNA polymerase will never see place to connection. Proteins will be absent. If we have mutation in part of DNA where transcription factors must connect it with a promoter, we will have disease. The same if enhancer elements will have mutation. Connection of RNA polymerase will be weak and DNA synthesis will not occur. Is it clear? Thank you. You must know this in first course. If you want to study genetics in the second, please. And the fourth level will be organisms level, when hormones can regulate any processes. exactly, for example, secondary sex characteristics. It looks like this. So steroid hormone will connect it with a protein structure, and this protein structure will be connected with promoter and show RNA polymerase where the place for transcription. Is it clear? Thank you. So DNA replication or reduplication of DNA. It is semi-conservative process. It means that every copy of DNA contains old DNA strand and new daughter strand. New slide, yes? Thank you. Just look like this. Red, it will be parent strands. You see after replication, we have two strands, two copies of DNA, one of them red, old parent, and one of them purple, new daughter strand. What is a replicon? This is a part of DNA where replication occurs. In bacterial prokaryotic cells, it will be one replicon. For eukaryotic cells, it may be a lot of places for replication. All DNA must be replicated, but not in one time. So it will be a lot of replicons in one time, but it will be several times for replication. The leading enzyme is DNA polymerase. And there are more than 7 types of eukaryotic DNA polymerases are known. They produce different proteins, different sequences, and it's important for genetics because if absent RNA polymerase, it means biosynthesis will suffer. So this is the variant how maybe looks eukaryotic replicons. Place between them will be replicated too, but in another time. You see replication forks and enzymes that create these forks. It will be in another place, I think. Okay, I will find them after. So, replication fork. You see that one strand have direction from 3 prime, if we will see from right side to left, from 3 prime to 5 and from 5 prime to 3 prime end. And the new strand of DNA will begin from 5 prime, so it will be opposite to 3 prime, and this strand will be synthesis more faster because without stopping. But another strand must begin from 5 prime, and it will be not beginning of the strand, beginning of the fork. So we will see several fragments of biosynthesis. We call them Okazaki fragments. Each of them will have primer for beginning, and they have different names, yes. So leading strand without stopping, and lagging strand that contains Okazaki fragment. Can I change the slide? Thank you. These enzymes, DNA helicase, with this enzyme also topoisomerase, and enzymes that small proteins that will catch DNA and stop their connection together. Can I change this slide? Thank you. So this is for you see topoisomerase, green. You see helicase that destroyed bindings between nucleotides, hydrogen bindings. You see small proteins, single-strand binding proteins that attach DNA and stop reorganization of DNA molecule. And you see two DNA polymerase in leading strand. It walk faster. In lagging, it create several Okazaki fragments. And you see blue DNA primase for creating primers. No one polymerase, DNA polymerase, will not begin work without primer. Is it clear? All of this? Again, from what place? But for this picture, you see DNA helicase unwinds the double helix. This is green, green topoisomerase. Helicase, helicase will be blue, dark blue. It destroyed hydrogen bonds between nucleotides, adenine and thymine, guanine and cytosine. From this place, I think it will be better during lesson, but I try. So again, you know that our DNA is a double helix strand. So direction for one strand will be from 5 to 5 prime to 3 prime end, upper strand in our picture. Another, by rule of opposite direction, will be from 5 prime to 3 from 3 prime, to 5 prime, down strand in our picture. But biosynthesis will always go from 5. So helicase will begin from this point. Yes. So it will be here, and it cannot be here, because this will be 3 prime end. So helicase will go in this point, and in this place, at last, beginning of biosynthesis. In this time, this DNA polymerase here, this DNA polymerase will come in this direction. So when helicase will be here, in this strand, leading strand, DNA polymerase will finish, but this RNA polymerase will finish only this fragment and begin work in this fragment. So this strand will be leading because this process is beginning from 5' to 3'. If it 5, it means it will be 3, it will be 5. Do you understand about this? Thank you. So the next. Topoisomerase will lead to this right-handed helix will be lined without winding, and helicase will destroy these bindings. These small proteins will catch DNA to stop again connection of them. So fork is ready. Replication may begin. And first, it will be DNA primase that create primer in this strand or in this strand. I'm sorry, not this. Somewhere here. So primer will be some. Not much watch. What? Givens, what is the question? Givens said, you may ask me next time during the lesson. I will draw it for you. Okay? Givens? Thank you. So we continue. After that, DNA polymerase will begin synthesis. I think, thank you. We must continue. We have 10 slides and they are all important. Okay? So, topoisomerase and SSB proteins, their function, topoisomerase, participates on overwinding of DNA. And SSB proteins, single-strand binding proteins, they stopped the recovering of these strands. Is it clear? Thank you. The next. RNA primase for synthesis of primer. DNA polymerase for growth strand. And there are several DNA polymerase, like RNA polymerase. And very important enzyme DNA ligase for for connection Okazaki fragments after removal of primers. by endonuclease. Can I change the slide? Can I? Thank you. Stages of replication. The first, synthesis of primer. The second, synthesis of Okazaki fragments. It's for legume strand, yes. After that, primer must be removed by endonuclease, and after DNA ligase will connect Okazaki fragment. I'm sorry. And I'm sorry. And the next enzymes, just do screenshot. If you will have time, you will remember this. If not, you will remember about this in the second course, find this information and will read. The same. Do screenshot and save it for second course. And about this we will talk in the another lecture about levels of chromatin. So if you have questions, you may send questions me on mail or for your teacher. Givens, you didn't be on our lesson, so you can come to department on Saturday from 5 to 6 o'clock. I will explain for you this process, okay? Thank you. And open site, there are present mails of all teachers. My mail, Vladimir Ilyich, or not, he's not, and Anton Evgenievich. So for today, our lecture is over. I was glad to see you. Thank you for your attention. Have a good day. Goodbye. Thank you. Goodbye. Thank you. And to you too. Goodbye.